Welcome back. So we are trying to understand the antigen presentation, both endogenous and exogenous pathways. We looked into how the MHC structures are, both MHC 1 and 2, and also we looked into how these MHC 1 and 2 are involved in processing of those antigen towards CD4 or CD8 specific immune response, respectively, when they are processed through MHC 2 and 1 respectively. So, after understanding this whole biology of antigen processing, MHC variability, and all the stuff which are required for making a variety of different repertoire of antigen presenting structures in the human body, we will move on and see how this MHC variability or MHC in total can regulate the immune response, and to that extent, why these MHC are so important. Is there any way to study the MHC specific peptide molecules or MHC as such molecule in total? So in this lecture, which is lecture number 14, we are understanding the MHC molecules as platform for immune communication. So let's move on and try to see a very critical thing which is important to understand the MHC variation. So let's try to see the MHC1 variability first, and then we'll move to MHC2 variability. Since we know that there are two chains in the MHC1, alpha and beta 2 microglobulin, and in alpha also we see that alpha 1, alpha 2 and alpha 3, which is depicted here, is the area where the antigen is binding. So one can expect that there could be a variation exist in the alpha 1 and alpha 2 fold in order to give access to the antigen varieties. And therefore, when people started doing the sequencing of the amino acid pattern on the alpha 1 and alpha 2, they found that there are great variation exist in alpha 1 and alpha 2, while alpha 3 is mostly constant. So this is the residue of amino acid. So say there are 280 amino acid big chain, alpha chain, and the red one is showing the change in the amino acid level. Say in position, say for example 15, we have a different amino acid in MHC1 molecules. Also in 40, next to 40 residue, you have variation. Again, there are red dots here and there which allows change of the amino acid in that particular location. What it implies? It implies that when there is a change in amino acid at the particular site of MHC locus, it can bind to a variety of different epitopes on antigen, and therefore we see this variation of MHC1 could lead to variation of its binding ability, and indirectly it leads to variation in immunoglobulin chain as well, because indirectly the immunoglobulins generated post production of these antigen or presentation of these antigen through this MHC1 molecules or two molecules can raise an immune response differently. So this is all about the MHC1 variability. Keep in mind that MHC1 variability mostly looked upon towards the variation in the antigen binding cleft between alpha 1 and alpha 2, and these are a few of the mutations. So that's giving an idea that alpha 3 is mostly constant and there is no variation as such. So these are kind of typically constant across the different individual and or in the cell type, but there are specific variation in alpha 1 and alpha 2. Well, in other hand, the MHC2 variability is unique because again in this there are two chains, alpha and beta, and in alpha and beta chains we have alpha 1 and beta 1 where maximum variability exists, and there is the groove where the antigen can bind between alpha 1 and beta 1. When people did the sequencing of those amino acid around this alpha 1 and beta 1, they identified that mostly the variability exists in the beta 1 chain. So beta 1 is mostly a variable in that region. So most of the variation exists in beta 1. So there is an obvious concern that the beta 1 is the site for variation, and therefore during the gene recombination event, which we have discussed in our antibody VDJ recombination, these variations are largely exist in the beta chain, and therefore we can see that these beta chain variation can give access to multiple antigens at that site. So this is just a pictorial representation showing the variation of MHC 1 and 2 molecules in general. Let's try to understand about these groups actually. We are talking a lot about these groups where the antigen can dock and produce an immune response and all. How this variation in the loop can decide the binding affinity. So let's try to understand this group in general. So this is actually the binding group which we have discussed in a cartoon fashion now. So these are MHC 1 binding group and this is MHC 2. Let's try to concentrate on MHC 1 binding group. So MHC 1 binding group, we can see that this is a closed topology. So the things are kind of closed, and we can see that the peptides are in a closed conformation, and therefore we can say that this group is having a closed topology. The configuration itself is closed. The N terminus of the peptide is here and the C terminus is here, and then we have this peptide length here. Now one can obviously see that this groove, because of the group pattern, the size of the antigen which are docked into this site is always smaller, and therefore we can see that MHC 1 molecule can only dock 8 to 10 amino acid fragment peptide in this area. Particularly this P2 and P9, these are the grooves which are typically giving a very common site for anchoring these peptides properly in order to facilitate its recognition by the CD4 or CD8 molecules. Particularly in MHC1, CD8 molecules will come into picture. So these groups are very important, and the sites are also there. We can see this anchoring domains are also buried inside these groups. While in MHC2 class, we can see that this is very open. It's a kind of an open. So this gives a flexibility in the bigger frame. So these are open groove topology, not like a closed group topology. And therefore, we can see this is the N terminus of the protein peptide, and this is the C terminus of the peptide, and this is kind of stretched out, and there could be multiple binding areas or anchoring areas where P1, P4, P6, P9, these are just name of the anchoring domain of amino acids into that group. So this gives a bigger picture altogether. So we can see that the MHC class 2 or HLA class 2, we can say that MHC and HLA are same in nomenclature. The binding groups are big, so they can give an access to up to 20 amino acid or 18 amino acid in general. So there are big areas which can dock the bigger size of the peptide. And in research, by doing the research, it has been identified that the 9 area in the core is the key binding factors which allows the proper strength of the peptide here, and if there is any mutation in this domain, the peptide may not bind properly, and therefore if there is a mutation in the P9 domain, we can see that the peptide binding is not happening and the person is lacking the MHC2 specific immune response. Now these are all very dynamic processes in the human body, and just to quote an example, there are virus infection. For example, when we study the HIV virus, HIV is a human immunodeficiency virus which causes immunosuppression in the individual. So the people infected with HIV don't produce an immune response in a proper way. The cells which are responsible for doing this is T helper cell, a CD4 positive T cell population. So CD4 positive T cell population can bind to MHC2, which we have studied in the last class. And therefore, this group binding is important, and HIV virus actually known to perturb the CD4 concentration by some of the protein, viral specific protein. So the viral specific protein can downregulate the CD4 expression, and because of the downregulation of CD4 expression, we may not see the recognition of MHC2 bound peptides, and therefore person may suffer from immunosuppression. So there are a lot of complex biology exist when we see this in prospects to a different pathogen altogether. So we'll move on and we'll try to understand its biology in different prospects gradually when we go along the course. So let's try to see the issues or the importance of MHC in different prospects in human body, because how this immune communication, which is the title of our topic, is related to MHC. And one of the things which we will study again in detail, but just for the knowledge, is graft. When we are transferring the tissue material, MHC plays a very, very important role. For example, we have given here the 3 examples. The first one is the skin graft to a syngenic recipient. Syngenic stands for the identical mice species. So we can see that these MHC locus are identical in these 2 species, and therefore the organ or the skin or any type of tissue grafted into other individual from a syngenic animal is getting tolerated. That means we can easily transfer the tissue from the species who are expressing a common MHC1 molecule. While in case of allogenic, that means both the MHCs are different in those 2 mice species. If you can transfer the tissue from one species to other, the tissue may not be survived into that system, because these MHC molecules in the second mice is different, not A, this is B, and therefore the immune response will be generated against this MHC A, which is transferred to MHC B mice, and therefore there will be a rejection. So there will be a very rapid rejection when there is an allogenic graft. Allogenic graft stands for the graft which are coming from a different altogether species. So this is the second condition, and the third condition is skin graft to minor H antigen incompatible recipients. So we can see that these 2 are both MHC-A allelic mouse, but if we see here, minor H antigens are the polymorphic peptides which are derived from the self protein, and they differ between the individuals. So that means there are subtle difference between these 2 species in terms of H antigen. And therefore, even though they are coming from the same lineage, they are coming from the same line, they may not tolerate the skin or any kind of tissue grafted into the different species, different mice here. So when there is a tissue grafted from one mice to other mice, even though there is a slight difference in the H antigen, we see there is a rejection, although that rejection will be little slow as compared to allogenic. Allogenic will be very rapid, and in syngenic that will be tolerated. Now what does it tells? It tells about that this is a very critical phenomena in burning issues, or in case of any tissue damage or any kinds of an injury. People always do and think about grafting of the tissue from very close relatives and all. The whole purpose of doing that is, if it is close, probably you will try to match the HLA type or the MHC type, and therefore more closer the MHC molecules, better is the chance of toleration or tolerance in order to hold the graft into the body. And therefore studying MHC biology is very, very important when it comes to graft biology. So we'll study this one in detail, the different types of graft host reaction in our later classes. This is just to give a picture that MHC does play a very important role in multiple processes. One such process is graft rejection. All right, we'll move on. So next one is the MHC does play a very important role in tumor killing. So the tumor cells are killed because they are identified by the human cell as a foreign molecule. Now how it is possible? Because tumor cells are our own cells. These are identified as a foreign is because of MHC1 molecules. Now if the cell is normal, they can present the peptide over the MHC1 molecule, and this peptide could be self-peptide. When this cell is converted into a tumorous mass, the cell might produce some additional protein which were not supposed to be produced in the normal cell, and that is what the indication that the cell is converted into a cancerous mass. So let's try to see that. Suppose this cell become cancerous, they started producing couple of extra proteins on their surface which were absent in the wild type or in the native cells, and therefore these are acting as an antigen. And if they are presented through MHC1 molecule, they can be identified as foreign in nature, because body thinks that these are non-self, these are not expressed by the normal cell, and therefore once the peptide against these proteins which are odd in tumor cell is processed through MHC1 molecule, the T cell receptor can identify it, and once identified as a foreign, then the cytotoxic T cell will produce lot of cytokines such as granzyme, interferon gamma, and other kind of thing, which can lead to toxicity in the tumor cell, or say killing of this tumor mass, eventually leading to a sterile condition in the human body. So this is basically an antigen-dependent tumor cell killing, because MHC is allowing the identification of those external peptides which are expressed in tumor cell and to be identified and destroyed by cytotoxic T cell population. Very, very important phenomena when it comes to tumor or cancer biology. Let's move on and see another thing which can give you an understanding of MHC importance. So this is just a pictorial representation of a research done for MHC1 dependent empty tumor activity. So what we understood, that there is a phenomena where a non-self peptide or non-self protein can be expressed by MHC1, and the cytotoxic T cell can identify this and destroy it selectively. Now there is a process where you can use anti-tumor agent, and anti-tumor agent also act via or through MHC1 pathway. So there is a drug called bleomycin, and bleomycin is a very well-known drug, anti-cancer drug, and what it does is, I have written here that bleomycin can bind to metal ions, and particularly the binding with iron or oxygen radicals or molecules which are present there, and they can form a complex that can cause damage to the DNA in a very specific way in order to generate reactive oxygen species in the cell where the bleomycins are added. Now, so therefore, bleomycin can rupture the DNA architect, and because of that there could be multiple phenomena happening inside the cell that could lead to anti-tumor activity, and therefore bleomycin is used as anti-cancer drug. How it does? So people have tried looking into the effect of bleomycin. So this is one pathway. What is the other pathway bleomycin does? So what we understood, that bleomycin once given to the patient is penetrating through this cell which is kind of a tumorous in origin, and these bleomycin can be taken by the tumor cell. There is a damage in the DNA, and because of the damage we can see that there can be expression of multiple kinases. So these are just name of some kinases. We will not go in detail about that, and these kinases can remove some activity so that NF-kappa beta or NF-kappa B, which is a transcription factor, will be free to do an activity on MHC1 gene. So what it implies, that when there is a bleomycin entry, there is a rupturing of the DNA which can lead to kinase activity, in turn leading to the transcription factor upregulation, which can do an upregulation of the gene transcript of MHC1. And more MHC1 will come, more and more peptide will be presented through MHC1 molecule, and therefore more and more T cell will be generated. So eventually the concentration of T cell will be 10-fold, 20-fold, 15-fold higher. Why? Simply because the tumor antigens which are odd will be presented through this MHC1, overexpressed MHC1, so that more and more T cell can be attracted to the sites. And once the T cell is attracted, more and more cytokines such as interferon gamma, and one such factor is granzyme B. Now what is granzyme is written here, we will study it later. So this is actually a serine proteases which are secreted by cytotoxic T cell, because of course we are talking about MHC1, so this is cytotoxic T cell. And they can very specifically do programmed cell death in a tumor cell mass, because essentially we can see that these basic molecules are having typical pattern where a perforin specific proteins are getting upregulated, and these perforins are assembled at the level of cell membrane to form a pore. So therefore perforin is because of the pore forming ability of a protein, and this granzyme can facilitate the diffusion of the ion through these perforin or pore which are produced by these proteins, and therefore there is a leakage of the content, and there could be a death of the cell very specifically when there is a granzyme B production in the system. So this is of course a very protective way to kill the cell, and when there is a bleomycin, this intake of the bleomycin can lead to this kind of pathway. So we can understand that even there are drugs available in the market which can perturb or which can modulate the expression of MHC1, which can lead to multiple different ways to kill the cancerous mass. We can think that Why we are talking about MHC1? Because these peptides are endogenously manufactured. Once there is an endogenous manufacture, we can always talk about MHC1 molecules. Well, all right, we'll move on and we will see the other activity which we have studied, and of course this is again repetition of the same which we have discussed. So we have discussed that dendritic cell can present the exogenous antigen through MHC2. We can also see that if there is an expression of MHC1, they can also present the endogenous antigen. They can activate either CD8 specific T cell when there is an MHC1 involvement, or they can also express CD4 specific immune response when there is an MHC2 involvement. That means these all pathway can lead to antiviral activity, or this all activity can help to cause destruction of the thing. So particularly here it is listed that CD8 specific immune response can give an anti-cancerous activity, while CD4 specific immune response can give antiviral activity as well. So why it is happening? Because these all processes are synchronized. They are all working in a very synergistic way in the human body. So there are cytokines which are produced by the T cell, which are called interleukin 2, and these are very critical cytokine which can act as a cell growth factors, driving the proliferation of and differentiation of different cell types in the human body, and can do multiple effects. IL-2 is known as biological mitogen. So we could act, we can get the IL-2 specific biology to proliferate the T cell population in order to enhance the immune system. So therefore these are biological mitogens. So they can enhance the production of different cellular mass. But how it is produced is because once we get an activation through this MHC pathway, we can get these all things done in a very routine. So this is what we can see, that these MHC molecules are critical in multiple ways to modulate the immune response. Well, how to study these MHC bound peptides? That's a critical thing. So what we know that this MHC is docking the peptide. We have understood the ways the MHC are variation in MHC1 molecules or 2 molecules, but we don't know how to study the peptide, because that's critical, because unless until we know that what are the peptides which are bound to those MHC, we can't study much on that. So there is a method called immunoprecipitation, and immunoprecipitation is a way where the immune cells are utilized to precipitate the antigens. So immune cells are actually these immunoglobulins. So what's there, that immunoglobulins will be used in order to do this process, so that we can understand the how these peptides and where the peptides, what are the sequence of the peptides, how these peptides are different than the other proteins. We can study those things together. Now there is a process. So 1, 2, 3, 4, these are all the ways one can understand this thing. So the first step is we can incubate the antibody with some of the proteins which are isolated from bacteria. These are the 2 proteins which are listed here, protein A and G. These proteins are uniquely identified from bacterial population. The protein A is coming from Staphylococcus aureus and protein G is coming from Streptococcus species. So these are all bacterial origin. What's the beauty about these 2 proteins which are coming from bacteria is they can bind very specifically to the immunoglobulin. So for example, this is heavy chain, and then we have a light chain, and they are joined by the disulfide linkage with each other. And here the protein it can be docked into the constant region of this antibody. So this protein A and G can be docked into that site. This is just a pictorial representation. The site may vary based on the different kinds of immunoglobulin, but mostly this could be an IgG molecule. So protein A and G can bind to IgG very specifically. People have looked into to make a hybrid of A and G, because A is having some specificity, so A might have a specificity with some species, G might have some specificity with other species. So if you can make a hybrid such as protein A/G, this is a hybrid form which can bind to a variety of different immunoglobulin G, and that can be utilized for this process. So this is just a process of precipitating anything that can bound to antibody. And people have used magnetic beads on those proteins so that this could be added with a magnetic sensor or magnetic particles, so that this might be helping them to elute out or precipitate out in a proper way. So in the first process, what is done is antibody, particularly the IgG, can be incubated with all those protein A or G containing magnetic beads, and we can see that these are all very specifically bound immunoglobulins, and then we can have a washing step where the unbound protein A and G can be washed out, and we can always have a very specific magnetic bead bound immunoglobulins. Then we can add the samples here. A sample could be a protein which we want to study, that what are the things which are actually binding to the immunoglobulins. So one can easily put the proteins of choice, and then we can incubate. Some of the protein can go and bind to the immunoglobulin, rest all will be eluted out, and once you unbound protein can be eluted out, we will have a very clear cut a magnetic bead bound protein where the immunoglobulin can be tagged with this peptide specifically. And of course, we can use magnet to pull down those all magnetic beads here, and rest all can be washed out. And in final step, we can elute the protein. So what we can do is we can increase the pH of the buffer, and then we can use to elute it, because we can increase the acidic pH based biology where the protein can be eluted out, and these proteins are very specifically bound to this immunoglobulin. So we could study this protein which can bind very critically to this immunoglobulin G. So therefore, this is a very important method, and we will see how this immunoprecipitation method can be utilized to do something called MHC typing. So this is what is MHC immunoaffinity assay, which is using the immunoprecipitation method. So let us try to understand this assay. So we have this dendritic cells or antigen presenting cell containing this MHC1 molecule along with the peptide, and we wanted to study this peptide, how these peptides are behaving, what are the biophysical characteristics of the peptide, biochemically what are the amino acids which are present in this peptide, whether these peptides are really doing anything. We need to study those things, and to understand those we need to know what are the amino acid sequence in the peptides or peptide chain. So once we have this sensitized dendritic cell which can be loaded with the peptide, we can lyse it. Lysis stands for rupturing the cell content, so that the lysed form of the cellular fragmentation can give out this HLA immunoglobulins. So lysed form of the thing can give this HLA bound peptide properly, and then we can do HLA immunoprecipitation. So we will have all the precipitation exactly like what we did immunoprecipitation. We can do HLA precipitation based on the immunoglobulin. So we can have the immunoglobulin that can bind to very specifically this HLA type, and finally we can have those peptide bound fraction with all the magnetic beads and all, which we had discussed in the last slide. By immunoprecipitation, we know that these are all the peptides there. We can do a peptide elution by increasing the pH, and once these peptides are eluted, we can do LCMS, which is liquid chromatography and mass spectrometry, which can use the biophysical as well as mass to charge ratio in order to elute the protein in a proper way, so that we can study properly that what are the things which are present in the peptide. So we can say that this is P1, this is P2, this is P3, and this is P4. So these 4 peptides are run on the details, and then we can say that, well, these are the characteristic feature of peptides through this LCMS analysis. So these peptides can be identified after elution, and once these peptides are eluted, they can be run over the LCMS, which is the liquid chromatography and mass spectrometry, which works on biophysical as well as mass by charge ratio, and this can be eluting the peptide in a proper way. We could study this peptide in a very sequential manner, so that we can identify what is the amino acid heterogeneity in between the peptide. Now, since these peptides are bound to the MHC molecules, we can say that these peptides are actually the peptide which are immunogenic in nature. because this can raise an immune response. And people use this as an epitopic structure, and we can map the antigen by these epitopic fraction in order to raise an immune response critically and to study the immune system in a proper way. So this is one such method where the peptides can be studied very critically using immunoprecipitation assay. So this is all about the MHC1 pathway, MHC2 pathway, and how these MHC molecules are important in order to do a variety of function in the cellular system in order to raise an immune response. So we will try to see something different in our next class related to immune system. Thank you.